Good afternoon, everybody. I'm Nicole Soranzo. I'm head of the Genomics Center, Human Technopole in Milano, New Life Science Institute, and also a senior faculty at the Sanger Institute. It's really a great pleasure to introduce this session after this morning session. So the session will start from now until a coffee break, and it's a CellMap session that already Ben has introduced this morning. For those of you who missed Ben's introduction, the section is really about developing knowledge, projects, and frameworks for studying cell function at scale. Function defined broadly, not only in terms of transcriptional and multiomic landscape, but particularly the expression of proteins and mediated function of genes, and also of attributes such as, for instance, the morphology or different types of cellular phenotypes that also constitute proxies for function. And really the idea here is that we want to begin to deepen and systematize our understanding of how cellular programs are controlled and enacted in individual cell types. So with this, it's my great pleasure to introduce the first speaker, the keynote speaker, David Valen. He's going to be joining, he's going to be giving his talk remotely. So the talk is recorded and he'll be joining later for Q&A. So David Van Allen is an assistant professor in the Division of Biology and Bioengineering at Caltech. Before becoming faculty, he studied mathematics and physics at MIT. He studied applied physics at Caltech, medicine at UCLA, and bioengineering as a postdoctoral fellow at Stanford University. At Caltech, his research group develops new technologies at the intersection of imaging, genomics, and machine learning to produce quantitative measurements of living systems with single-cell resolution. So it's a great pleasure. It should be a very interesting talk, and very much looking forward to it.
Greetings, everyone. Thank you for the invitation to speak. Today, I'll be sharing my lab's work at the intersection of biological imaging and deep learning. I would like to start this presentation with a 30,000-foot overview of the intersection between deep learning and the life sciences in general. The way I like to think about this intersection is in terms of the different types of data that are collected about living systems. Broadly speaking, there are three types of data that we typically collect. There's imaging data, which contains information about the spatial and temporal variation of living matter. There's sequence data, which contains information about the parts list variation of living matter, which genes are present, DNA sequencing, which genes are turned on or turned off, RNA sequencing, and whatnot. And then there's structural data, which contains information about how biological molecules interact with the physical world to give rise to biological form and function. Over the past few years, for each of these different data types, there's arisen a collection of deep learning methods that can do quite remarkable things with these data. For imaging data, these are convolutional neural networks, which feature quite prominently in today's talk. But be aware that there are other flavors of deep learning methods for these other data types as well. For sequence data, there are language models that can perform equally remarkable feats with sequence data. Here, we have a figure from one of my favorite papers of the past couple of years, from Brian Bryson and Brian He, published in Science in 2021, where they showed that they took a language model and trained it in a self-supervised fashion on a large collection of viral sequences. What fell out was a deep learning model that was able to accurately predict which mutations enabled viruses to escape detection by the immune system. For structural data, these deep learning methods include equivariant graph neural networks, flavors of graph neural networks which respect relevant spatial symmetries like translation, rotation, and whatnot. These equivariant graph neural networks are key features of the deep learning models that have made transformative advances in protein structure prediction and protein design. The previous slide provides the context within which my lab's work takes place. Zooming in more closely on imaging data, it's fair to say that new deep learning methods have drastically changed the relationship between life scientists and imaging data by making it easier to access quantitative information that's present within images. A collection of tasks, which used to be quite difficult, can now be performed fairly routinely are shown here on the slide. This includes tasks like cell segmentation, identifying individual cells and images, cell tracking, linking cell detection events together over time and identifying division events to construct cell lineages, identifying single molecule spots in RNA fish imaging data, as well as some more imaginative applications such as restoring noisy images and augmented microscopy, inferring fluorescence images from label-free images. Concurrent with these advances in algorithms, we have also had advances in imaging technologies that have substantially increased the information content of cellular images. These include advances in reporters, which capture information about different physiological processes in single cells. Advances in multiplexing have led to new imaging platforms that make it possible to image dozens of different protein species and thousands of different RNA transcripts in the same sample. Last, we've also had advances in resolution, which refers to the collection of super resolution imaging methods that allow us to beat the diffraction limit.
These two advances in combination create the possibility of images being a universal data type for biology. One advantage of this vision is the ability to integrate information across measurement modalities while conserving cellular identity. Let's say we wanted to measure signaling and morphology dynamics and then measure the impact on gene expression for 1,000 genes. To do that, we could simply perform live cell imaging of a cell line expressing a relevant live cell reporter, fix the cells after imaging, and then perform an endpoint spatial transcriptomics measurement like seqFISH or MERFISH. Because all the measurements were performed with imaging, we have a natural way to conserve cellular identity across these different measurements. Advances in commercialization in spatial genomics are making these types of measurements more accessible. A major challenge that is ahead of us is the construction of computational primitives, key algorithmic building blocks that can perform essential computer vision operations like cell segmentation or cell tracking seamlessly across diverse biological imaging data sets. In this slide, I would like to provide a concrete example of what I mean by a computational primitive. Here we have a classic cell biology movie of an immune cell, a neutrophil, chasing a bacterial cell, finding it, and then eating it. We would like to create a deep learning system that can produce a narrative about this imaging data, very much like what I just described, with entity-level resolution. We can go through a thought exercise of what such a system would look like and what it would need to know. The very first thing you might want the system to know is the notion of what's a cell. It should be able to identify cells in each frame and know when these detections are referring to the same cell or different cells. It should also be able to understand the semantic or biological layer of information as well. It should know the notion of cell types, that these are red blood cells, that this cell is a neutrophil, and that these wiggling black dots are bacterial cells. And last, it should be able to understand interactions between cells. It should know that there are no meaningful interactions between the red blood cells or any other cell type in this movie. It should know that at the beginning of this movie, there's an interaction between this neutrophil and this bacterial cell called chemotaxis. And then it should know that towards the end of this movie, this interaction changes into phagocytosis, and that when this phagocytosis event happens, there's a change in state in the bacterial cell from being alive to being dead. Essentially, what these computational primitives are meant to do is convert these raw imaging data into a more interpretable format, which is the spatial temporal graph, where each entity in this movie is a node in this graph, and you have node level properties like cell types, and then edge level properties denoting the cell-to-cell interactions. By specifying things in this way, immediately we can see where the different computational primitives might come in. We need a computational primitive for cell segmentation, identifying these cells. We need a computational primitive for tracking cells over time so that we know which cells, which detection events refer to the same cell. And last, we need computational primitives for identifying cell types and then also identifying the cell-to-cell interactions. If robust and general computational primitives are what we need, then the natural next question is how do we make them? This gets into the title of today's talk. While deep learning methods are essential to get them to work in practice, you need much more than just deep learning. You need data. This includes representative data models should perform well on, in addition to the labels they need to learn from. You need the deep learning models, and then you also need the computational infrastructure for both model training and deployment. In our view, these three challenges should be addressed simultaneously rather than approached in isolation so that one can harness the synergies that exist at their intersection. The challenges in these domains and their intersections can be viewed as primarily software engineering problems, which is the reason for the title of this talk. Everything in this space can be viewed as code.
With the introduction out of the way, I would like to share a few stories that we've been working on in my lab. The first story centers around single-cell analysis, the problem of finding single cells in images. In our wet lab, we use live cell imaging to understand how cells process information about themselves and their environment. In these experiments, cells express a live cell reporter, where changes in the amount and location of fluorescent signal reports on the activity of a signaling pathway. To interpret these data, we need to first identify cells in every frame and then link these detection events together over time to create temporally consistent records for each cell. We can then query each cellular compartment for its brightness to convert these raw data into a quantitative interpretable format shown here on this slide. Single-cell analysis is also a challenge for tissue imaging experiments. New experimental platforms can measure the location and abundance of dozens of different proteins or thousands of different RNA species simultaneously in tissues. These methods essentially produce n-color images where n is a number between a dozen and 1,000, and each color corresponds to some biologically meaningful variable. Here we see some example data taken from a paper by Michelangelo and Liat Karen published in Cell several years ago, where they profiled the tumor immune microenvironment in a cohort of patients with triple-negative breast cancer. These measurements produced rich data for each patient, but analyzing them is challenging. One of the first steps in analyzing these data is identifying which pixels belong to which cells. If you can do that, you can query the brightness of each color, perform clustering to identify cell types and states, identify spatial patterns, and if there are outcomes data, link these patterns to outcomes. What is needed in both of these spaces are computational primitives. For live cell imaging, we need primitives that can segment cells, track cells, and construct cell lineages for commonly imaged cell lines. For multiplex tissue imaging, we need computational primitives that can reliably perform nuclear and whole cell segmentation across all the different imaging platforms in use and all the different tissues being imaged. To enable the construction of these computational primitives, our lab has been working for the past several years to create an ImageNet for cellular imaging. We've compiled a large collection of tissue images that include both a nuclear stain and a membrane or cytoplasmic stain. We have also assembled a similar collection of live cell imaging movies collected with phase and fluorescence microscopy across a collection of commonly used cell lines. While data and metadata ingestion is ongoing, the largest challenge ahead of us is annotation. This includes the difficulty of labeling each image and the problem of scale, having too many images to label. I would like to show you some of the raw imaging data to provide a sense of difficulty of the labeling task. For tissue imaging data, a major challenge is cell morphology, which varies substantially both across and within tissues. The signal-to-noise and autofluorescence characteristics also vary across platforms. Some cells do not have a cell nucleus, which is an artifact of doing two-dimensional imaging of three-dimensional objects, and in some tissues, cells are in very close proximity. For live cell imaging data, the challenge is time. Entity-level labels must be temporally consistent, while dynamic events like cell divisions and cell deaths must also be labeled. In addition to the difficulty of labeling each image, there is the problem of scale. We simply have too much data for humans to label manually by themselves. The approach we have taken is to have humans and AIs work together to solve the labeling problem. The challenge then is to create software systems that enable this type of collaboration. We have built such a software tool, Deep Cell Label, to enable exactly this type of interaction for biological imaging data. The software tool is browser native and allows distributed labeling of large biological image collections. Here we see a user labeling division events in live cell imaging data. Other modes of this tool allow for similar sorts of labeling for tissue imaging data. With Deep Cell Label, we have also thought about how human-AI collaboration can accelerate the labeling of large image data sets. In a paper published in Nature Biotechnology earlier this year, we described a human-in-the-loop approach to image labeling where crowdsourced workers correct model errors rather than create labels from scratch. This approach reduces the marginal cost of labeling as the project proceeds. More data leads to more accurate models, which make fewer errors that require correction. We demonstrated this approach can substantially increase the labeling throughput. We use this approach to label a large collection of tissue imaging data. The resulting dataset, TissueNet, contains over 1 million paired nuclear and whole cell labels from 6 different imaging platforms, 9 different tissue types, 3 different disease states, and 3 different species. Labeling this dataset took 4,000 person-hours or 2 person-years worth of work to create. Because it was done in this distributed fashion, it took substantially less than 2 years to complete this project, and most of the labeling burden was carried by the crowd. The time spent by experts was primarily on crowd management and quality control. With this large labeled data set, we created a deep learning pipeline called Mesmer for simultaneous nuclear and whole cell segmentation of tissue imaging data. Mesmer's best feature is that it generalizes across different tissue types and imaging platforms. Here we see a collection of different tissue images. And here we see whole cell segmentation predictions produced by Mesmer, along with the associated F1 score. We found Mesmer to be so accurate that when we compared its performance to human performance, we found that it was able to perform whole cell segmentation with an accuracy that was on par with what humans can do. You can find more information about this analysis, as well as information about downstream analyses that Mesmer enables, including cell type detection and quantifying cell morphology, in our paper in Nature Biotechnology. We have been hard at work building a similar resource for live cell imaging. Here, the major challenge is time, as we need to ensure entire movies are labeled rather than just static frames, and we also need to make sure that the labels generated are temporally consistent. The size of our labeled data for live cell imaging is quickly approaching the size of TissueNet. The advantage of labeling entire movies is that we can develop models for cell tracking and lineage construction in addition to models for cell segmentation. Our approach treats the tracking problem as a classification problem. Given a cell in frame I and another in frame I+1, we determine whether these are the same cell, different cells, or if there's a mother-daughter relationship. Our approach uses graph neural networks to integrate information like appearance, location, and then the neighborhoods around each cell to create features that allow us to maximize performance on this prediction. Using Deep Cell Label and this new tracking model, we have been able to crowdsource the annotation of entire live cell imaging movies. These large data sets lead to accurate cell segmentation and cell tracking algorithms. Example results are shown here on this slide. Visually, you can see that these models can accurately track cells and identify division events. The next computational primitive we've been working on is the spot detection algorithm for single molecule RNA fish experiments. RNA fish involves fluorescently labeling mRNA transcripts, which allows you to measure gene expression while preserving spatial information. Multiplexed single-molecule RNA fish measurements, like seqfish and merfish, can measure the abundance and location of thousands of genes in the same sample. This is achieved by labeling the same transcript multiple times in sequential imaging rounds, which creates a combinatorial barcode. In this example, transcripts are labeled with one of 4 colors across 6 different imaging rounds to create a barcode that corresponds to a particular gene. The analysis of these multiplex single-molecule fish imaging data requires robust image alignment, cell segmentation, and most importantly, spot detection. Approaching the spot detection problem with supervised deep learning methods is challenging because of the data. Most single-molecule fish data sets contain too many spots for humans to curate manually. We have taken an alternative route and developed an approach to training deep learning models for spot detection with weak supervision, inspired by recent work in task programming. In our approach, we fine-tune a collection of classical spot detection algorithms to a training data set. Each image has a set of labels, one from each classical algorithm. We then perform generative modeling to find consensus labels and train a deep learning model for spot detection on these consensus labels. Our benchmarking has demonstrated that this approach provides superior performance to models trained on either simulated data or data derived from a single classical algorithm. Here we see predictions on a raw image of which pixels contain a spot, as well as a regression prediction which allows us to obtain subpixel resolution. Simple thresholding on the classification prediction image then provides concrete spot predictions. We have taken this computational primitive for spot detection and combined it with our computational primitive for cell segmentation, as well as a graphical model that decodes gene identities. The end result is Polaris, which is an integrated pipeline for multiplex single-molecule fish imaging data that requires no manual parameter tuning. Here we see the output of Polaris on merfish data from the terminal mouse ileum. We believe that Polaris will alleviate a major bottleneck that currently exists for spatial transcriptomic imaging data. The methods I have described today are only useful if they are shared widely. Last year, we published a paper in Nature Methods describing a software package called Deep Cell Kiosk, which enables dynamic scaling of deep learning-enabled image analysis pipelines in the cloud. The scaling is dynamic. As more data is sent for processing, the software recruits more computational resources to minimize latency. The software currently powers our web portal deepcell.org, which shows several of the models I described earlier in this talk. In my last few slides, I'd like to give you some thoughts on the future direction of our work. Looking at cellular image analysis methods more broadly, the way in which they differ the most is in how they represent the idea of what's a cell. A collection of different representations are shown here. They include the interior border background pixel representation using the original UNet paper, as well as the deep watershed representations used in our work. Currently, different methods choose representations that are best suited for a given data set. While this approach has been successful, it poses a challenge to creating universal models that can operate across different data sets. Moreover, there are challenges innate to this representation engineering. One challenge that exists are overlapping cells, which are quite common in live cell imaging movies. Representations that impose a constraint that each pixel must be uniquely assigned to a cell cannot perform well on these imaging data. This constraint has impact throughout the entire software stack. Removing it requires changing the labeling software, the label formats, as well as improving the deep learning models so that they can predict sets of cell masks directly from imaging data without passing through an intermediate representation. Over the past several years, we've been exploring transformer-based models for cell segmentation, which have the potential to remove the need for representation engineering. Here, we have preliminary data showing that a transformer-based cell segmentation method can perform well across a diverse set of images without the need for an intermediate representation. While much work remains to make this preliminary result into a useful software tool, these early results are quite promising and provide hope for the construction of a foundation model for cellular image analysis. That concludes this talk. I would like to thank my lab members who worked tirelessly day and night to create the results that I presented today. I'd also like to thank collaborators, Michelangelo, Liat Karen, Long Kai, Yi-Song Yu, and Eric Osterman, as well as several different organizations for funding and support. Thank you.
Thank you, David, for a fantastic talk. I think there are questions online, so we might start with those, but please raise your hand if there's also a question in the room.
Yeah, thank you. So via Slido, someone's asking sort of how many single cells are required for these deep learning approaches?
Depends on the accuracy that you're trying to achieve. I would say we roughly, for new models, we try to get at least 10,000. Once you're around 100,000, you start getting these, performant models. Past that, you start, around the, I'd say 250,000 to 1 million number, you start to get the human level performance type models. We have yet to see a case where the models perform worse with more data. Although, as you start getting out, past 10 to the 6, you start getting diminishing returns. The Nature Biotechnology paper, we described the construction of TissueNet and the development of Mesmer has an analysis of accuracy as a function of data set size. We'll explore this in more detail.
Are there any questions in the room?
Do you have another one online?
Otherwise, I'll take another one from our online folks. So someone's saying that in histology analysis, it looks like labeling of the data or using training data sets is often not better than using unlabeled strategies. Is that the case for your experiments as well?
I think it depends on what you're trying to do. And so if your analyses can be done with image level or patch level labels, there are self-supervised or unsupervised approaches that can mitigate the amount of the need for large numbers of labels. For here, really what we're trying to do is single cell analysis, getting the models to recognize, that unit of organization that's present in the images. And there, just the morphology of cells differs so much, within tissues, across tissues, in different contexts, live cell imaging, tissues. It differs, quite substantially across different species, even, right? If you're looking at bacteria versus yeast versus mammalian cells and the different image modalities, we personally just felt that, if you wanted to make progress today, it would be easier to tackle the labeling problem rather than come up with some super clever self-supervised or unsupervised approach to this problem. And so, is there some label-free or approach that's out there that could potentially meet this need? it's possible, but it's not there today. I will say, like, there are methods for reducing your label, the reducing the number of labels you need to achieve a certain level of performance. And in some contexts, we explore those in our lab. So like as I mentioned before, self-supervised learning, contrastive learning, you can generate embeddings and representations, where if you use that as a way of pre-training models, you can shrink the number of labels you need by an order of magnitude or so. We use those in different contexts in our lab, but nonetheless, you still can get a lot of value from this infrastructure for labeling large data sets in a way that really lets you pull the most information out.
So David, I had a question too. So thinking about what we're trying to do here, which is genetics on populations, among other things, do you see value in trying to harness some of the wealth of data that is potentially sitting in biorepositories, for instance, imaging data, even historical archives of slides taken by hospitals over time? What are the technical challenges you see from doing that, from maybe loss of staining to other things? And also, if you were to think about experiments moving forward, would you think that probably prospective sampling around specific projects is a better strategy compared to a generic harnessing of data that exists out there?
Yeah, I think that's a really good question. I think broadly speaking, I kind of view this as a genotype to phenotype mapping, right? You've got some space of genotypes that you want to map into some space of phenotypes, and imaging is one modality that you can use to phenotype living systems. Say broadly speaking, there's two different avenues you can one might take. You could imagine doing some of the more modern multiplexed imaging of fixed samples, or you could do live cell imaging if you have some cell lines that derive from patient samples. And there, I think it really depends on what phenotypes are you expecting from the genotypes that you're interested in. You could go. There are scenarios where you could use either. I'd say, like, intellectually speaking, we're getting more interested in the amount of information that can be pulled out of dynamic live cell imaging assays, both of the label-free variety, which are fairly straightforward to perform, as well as the ones where you put in a suite of fluorescent reporters and use that to report on the activity of different physiological processes. I think the challenges there are I'd say, like, the biggest ones I kind of foresee are more like logistics, getting samples preserved in such a way where the folks who are best suited to do these sorts of measurements can get ready access to the samples they would need, having all the associated metadata, so that if you're imaging a sample, you have a good idea of the genetic background that it came from, and then making that a data that flavor of data available to the folks who sort of more on the machine learning side. In our lab, we do both, but broadly speaking, that's there is this this separation right now between the two fields, and that's something that I kind of foresee being like being a challenge. I would say, like, there's also while there's one path you can take where you're harnessing sort of the natural genetic variation of populations. There's another world where you're doing these deep mutational scanning experiments, and you have, like, clever ways of sort of matching the mutations you're inducing to the phenotypes that you're seeing on the microscope. So here, I'm thinking of the work of Paul Blaney. He's a pioneer in this space. There's also work on doing, sort of pooled intron tagging, tagging different genes with fluorescent proteins in a high throughput pooled fashion. These flavor approaches, I think, also have could potentially have utility here as well. So that's kind of what I'm seeing. I think the biggest challenges are at the beginning are just going to be logistics.
Thank you so much. Lots of food for thought. I think if we don't have any more questions, I'll move on to the next speaker. Thank you very much, David, for joining us.
Thank you, and looking forward to the panel. See everyone in about an hour.
Okay, so it's our joint pleasure to introduce our next set of speakers. This is a double act. Gosia Trynka and Melina Claussnitzer will be jointly giving both a keynote and a pitch presentation in a configuration. So I would like to first of all introduce Gosia Trynka, who's been a colleague for many years at the Sanger Institute. She's the head of the Immunogenomics Research Group and also the Director of Experimental Science of Open Target, this platform including private and public representation, seeking to use genetics to advance drug discovery and development. Before this, she did a PhD in Groningen, working mostly on celiac disease, and then a postdoc at the Broad Institute working on developing computational methods for, particularly for enrichment in immune genetic diseases.
And it's my pleasure to introduce as well Dr. Melina Claussnitzer, who will be speaking with Gosia in the upcoming talk. Dr. Claussnitzer is an institute member at the Broad Institute and an assistant professor at MGH and Harvard Medical School. She co-directs the Type 2 Diabetes Systems Genomics Initiative, and she serves as associate director of the scientific strategy for the Novo Nordisk Foundation Center for Genomic Mechanisms of Disease at the Broad. Her lab focuses on the dissection of the genetic basis of T2D and its comorbidities into actionable therapies. Dr. Trynka and Dr. Claussnitzer are the co-leads of the CellMap working group of ICDA.
Great. Thank you very much. It's a pleasure to be here and pleasure to share the stage with Melina. We thought that really by doing this double act and together with David, we would set the stage appropriately for our pitch later on. So what I'd like to do in the next 15 minutes is focus on one of the stories that we recently published from the group that looks at one of the approaches that we've undertaken to link immune disease variants to target genes and cellular functions. So this is really to address what pretty much we're all are here for, which is that we're getting these associations for diseases for human phenotypes. And the next step is to really translate those associations, those genetic variants to function. And first of the approaches in this roadmap is understanding which are the disease-relevant cell types. And once we're landing in the right cell type, then we can start asking questions about more of the mechanisms that are underpinning these disease associations and the cellular functions. So we can ask about which are the disease relevant genes, the causal genes, and which cellular functions they perturb. And really these two are critical because this is where therapies work. They work by actioning on the cellular biology. So it is really critical that we get better understanding of how genetic variants are regulating cellular functions. For immune diseases, where there's a wealth of GWAS associations, we've known now for years that one of the critical cell types for immune diseases are CD4 T cells. And one of the predominant roles of CD4 T cells is to recognize a danger in the body, undergo activation, and upon activation, perform its effector function. So once the T cell is activated, it starts to proliferate, undergoes a number of divisions. Depending on the environment in which it is, it will then acquire different effector functions. So it will become a helper cell of different roles. And really, the roles are dictated by the types of cytokines that they secrete and the types of molecules that they have on the cell surface, how they interact and who they interact with, and so on. And obviously, this is very cellular granularity, and one cannot get at these questions easily by performing bulk type of approaches. So what we did in our study was to recruit 119 healthy individuals from whom we isolated naive and memory T cells. Naive T cells are cell types that haven't undergone previously activation, and memory cells have previously been activated, and they hold that memory, and the 4-day response to activation is slightly different to naive cells. But overall, they tend to be put into one shared bucket, CD4 T cells. They're fairly similar, yet they have quite distinct functions. And so using single-cell technologies, we wanted to understand better how these two similar yet different cell types how the programs in these two different cell types are regulated by genetic variants as the cells undergo activation. So for that, we sample cells at resting state at 16 hours, which is just before cells undergo division, at 40 hours, which is when cells typically have undergone division, and 5 days, which is kind of a late time point when the cells have acquired effector function. And by doing it at scale across over 100 individuals, we were able to then perform EQTL mapping. And the goal of performing EQTL mapping was that we could then look at the colocalizations with disease associations to get a better idea which are the target genes, in which cell types they are regulated, at which time points, and so on. So with this, this is our beautiful UMAP, and it is beautiful because you can really see a lot in this figure. So blue are naive cells and red are memory cells. The lighter shade of color is early activation time point, with the lightest one being actually resting time point. And then you see that the cells are progressing throughout this activation trajectory. And you can see, as I said, they're fairly similar, so they are clustering together, yet they are occupying separate space. So there's a clear distinction between memory cells in the inner part of the UMAP and the naive cells that are on the outside. Now, what we were able to do is identify different states of the cells. So as I mentioned, we sampled cells at 4 different time points. And one of the interesting observations that we saw was the fact that we could see this cluster of lowly active cells, and this was mainly coming from the cells that we sampled at 16 hours and 40 hours. So these are probably cells that we're capturing that's just been activated. They have distinct phenotypic, they have distinct transcriptional signature, so they're not yet 16 hours or 40 hours. And obviously we would have missed that by deploying a bulk RNA-seq approach. There's a range of different well-appreciated subpopulations of T cells. And actually, I'm not going to go into details on those. What I'd like to highlight here is that as we're looking at the different time points of activation, we were able to identify 38 distinct clusters, of which what was really interesting were the clusters that were appearing only upon activation. So for example, we saw this cluster of cells expressing interferon genes that occurred already in this slowly active cluster of cells and was maintained throughout all of the activation time points. We also saw a cluster of cells expressing NF-kappa B genes that was only present at 16 hours and 40 hours. And that's really interesting because that allows us to then identify features such as, for example, it's known that upon activation, T cells will quickly upregulate interferon genes. But we actually see that it's not that all the cells are upregulating interferon genes. We see that there's a cluster of genes that is very much prone to expressing those genes. So we've used this data set to map EQTLs, and by doing pseudo-bulk and bulking cells into broadly naive and memory categories and different time points, we identified nearly 6,500 EQTLs. And we were able to identify effects that were very much cell type specific, such as this one, IL-12RB1, where we see that there's an effect in memory cells, but not in naive cells. We were also able to identify effects that were very specific to individual cell states and cell types, such as the example of GFPT1 gene. more so, we also went and mapped the EQTLs in individual subpopulation, and we were able to get further insights into how genes are regulating expressions of genes in specific subpopulations and detect effects that are very much cell population specific. I'm not going to go into details on that one, because what I'd like to focus on more in this discussion is in this talk is really leveraging the dynamics, the time course of activation that we had in our data set. So we wanted to understand if we could map the dynamics of gene expression changes throughout the T-cell activation. For that, we performed a trajectory inference, and you can see that the trajectory starts in the resting state in here and then transitions throughout the activation time points. And to validate if this trajectory was really resembling what we would expect to see, capturing early, mid, and late activation time points, we ordered cells based on the coordinates in the trajectory map, and then plotted the expression values of the genes that are well known as the T-cell markers for different stages of T-cell activation. So here I'm showing you an expression of CD69 gene. Keep that in mind. Melina will come back to that later on. You can see that CD69 peaks very quickly in early time points of activation, and then it goes down in the later time points of activation. While IL-2RA peaks early in the activation, and then the expression levels are maintained at high levels throughout the activation. And finally, IL-7R decreases with the activation. So this was really encouraging that the trajectory was truly capturing T-cell biology that we know from decades of immunology studies. So using this trajectory, what we did is we ordered cell types for each individual into 10 bins, and then we used these bins to map the effects of genetic variation on regulation of gene expression dynamically. So what we're looking for is the effects where there are different effect sizes of EQTLs at different time points of activation, such as in this case, for example, you can see that the effect size increases as we progress throughout the activation time course towards the later times of the T-cell activation. So this is a high-level schematic overview of the approach. This is how it looked in practice. We were able to detect really all sorts of effects of gene expression regulation. So, for example, GBP7 has a very large effect at early time points of activation, but then it diminishes as the cells progress into later time points of activation. The gene in here has a very large effect at very early time points of activation, including in resting states, and then it goes down with the activation. Then we also pick up effects that are more linear, like this gene down here. Overall, of all of the EQTLs that we've mapped, we saw that for a third of the genes, we were able to identify this dynamic regulation of gene expression throughout activation. And interestingly, a lot of those genes that are dynamically regulated are enriched in processes such as cell metabolism or T-cell proliferation and division. So here's an example of a gene that is colocalizing with disease association. That was the ultimate goal of developing this data set. We've identified close to 130 different colocalization events. I'm not going to go through all of them. I just want to highlight CTLA4, because this is an excellent example of a gene that's very therapeutically known, because it is used in treating rheumatoid arthritis, where CTLA4 antibody is delivered to patients with rheumatoid arthritis, indicating that there's a CTLA4 deficiency. And what we see here in an example of colocalization with type 1 diabetes, but we also see this gene is colocalizing with celiac disease and rheumatoid arthritis, is that there's a strong effect in early activation of memory cells, such that the disease risk allele is decreasing the expression of CTLA4. And that is very much in line with this therapeutic application of CTLA4, which indicates a deficiency of CTLA4 in patients. And it is really interesting because the role of CTLA4 is to down-regulate activation of the immune system. So if you are deficient in the CTLA4 levels, therefore your ability to to reduce the activation of immune system could be reduced, therefore kind of following the line of immune inflammatory diseases. Now it's an also interesting gene because you can see that it is highly expressed in naive cells as well. However, the expression patterns are very different between the two cell types. So in memory cells, the dynamic effect, the high effect size that we see is in the early time points of activation. But in naive cells, this is really later on in the activation time course. So to wrap up, by looking at all the colocalization events, we actually saw that if we had only taken cells in a resting state, as majority of the studies often do, we would pick up about 40% of all the colocalization events that we ultimately detected. By adding in just one activation time point, we increased this number to 60%, but adding in 2 extra time points, we really add in additional 40%, highlighting how important it is that we start looking at biological processes dynamically at multiple time points of a cellular process that we're studying. And what's really interesting is also the fact that when we look at the number of genes that are colocalizing and express these dynamic effects, 60% of all the colocalizations have this dynamic gene expression regulation. But when we compare that to all the EQTLs in our data set, as I mentioned earlier, it's only 30% of the genes for which we identified the dynamic gene expression regulation, really indicated that disease-associated variants are enriched for this dynamic effects. So it's all great. It's getting us a bit closer to the disease-associated genes potentially, but I think still we're left with this question, but how do these genes really impact cellular functions? How do disease-associated variants impact cellular function? And this really is something that as a field we need to start thinking more about. So do these genes impact cellular morphological features? I mentioned earlier that we see that these dynamic effects are enriched for pathways such as cell metabolism or cell proliferation. And here's just an example of T cells as they progress through activation. They also metabolically change very much. So this shows you an increase on the density of mitochondria in these cells as they go from 16 hours to 5 days of activation. And metabolic activity is really important for defining effector cellular T cell functions. So we ought to get better at really getting from disease associations and genetic variation to cellular function. And with that, I want to thank all the collaborators on this project. Really want to acknowledge Blagoje and Eddie who led this work, and Julie and Carla who contributed with the cell imaging figures. And I'm going to hand it over to Melina now.
Super job. I have to briefly switch, sorry. Everyone can chat a bit. Let's see.
Again, just a reminder to our folks online, we did have about 80 folks join this morning remotely. Feel free to submit your questions at slido.com with the code 5066.
Super. Yeah, so it is such a great pleasure to be here and see again those ones that we last saw each other in DC, I guess, and had a wonderful time. And I'm also very, very much looking forward to meet new friends and colleagues and discuss the science we are running and the proposal that we are pitching later. It's a particular pleasure to double act with Gosia today and later on to pitch our proposal on behalf of the CellMap team. So I'm going to talk very briefly, over 15 minutes, about some of our work in the context of mapping cellular processes underlying type 2 diabetes genetic variation that we are particularly interested in our lab. And of course, as all of us know, the tremendous success in large-scale genetic association studies in type 2 diabetes have come up with more than 700 genetic risk loci that robustly associate with this disease, and even more from glycemic traits, and more than 1,000 for obesity-related phenotypes. So with that, this success really laid the foundation, I guess, for creating comprehensive and unbiased catalogs or maps of cellular processes, biological processes, right, that robustly link the genetic variation that we are looking at on the one side to diseases on the other. So it is a tremendous opportunity for our community right at our fingertips, provided that we can actually convert the genetic variation to their actual biological function. We have, not going to go into detail at all, but just very quickly defined this principled hierarchical framework from going from a genetic association study to the actual causal variant, its regulatory elements, its genes and proteins that are underlying the genetic variation, its cell states and cell types, and the cellular morphological and cellular functions that are related to the disease progression and disease manifestation. And the reason I wanted to briefly mention this variant to function model is that in our experience, the kind of establishing these causal relationship between each and every of these layers has proven to be really important in order to make it down to the real mechanism that is underlying a given genetic variation. So in our work initially, very briefly, we identified an intronic variant controlling long-distant two target genes, IRC3 and RX5 in adipocyte precursors, and having a cellular phenotypic effect on mitochondrial content and thermogenesis rate in adipocytes. And that kind of shifts this dynamic balance between energy-dissipating beige adipocytes to lipid-storing white adipocytes. We also worked with Marcelo Nobrega very closely on multiple projects over the last years, and one of them has been the FTO locus, where we are identifying additional non-coding variants that actually act in the hypothalamic neurons. That's, Marcelo's postdoc back then, Deborah Sabrera, has been beautifully driven this work and shows that the same target genes are controlled. Similar processes involving the mitochondria are regulated and lead to macronutrient selection in humans. So, this kind of model of going from variants to all the way down to biological processes and phenotypes seems to be informative. It also appears to generalize this framework for going from variants to the cells to the biological pathways. These are just a few examples, a snapshot of what we have been working on over the past few years, in either published or currently in review or in preparation. And I guess actually two notions. The one is it is not easy to connect the genetic variation, of course, to the genes. We all know that. But it is even more challenging to actually identify the cellular phenotype, and typically takes quite a while. And I will get back to this in a moment. The second learning that we had is that this process of a single locus variant to function dissection study is highly laborious, time-consuming, hand-to-hand type of combat work, where you really typically, as a lab, spend 5 to 7 years involving multiple funding cycles to start off a genetic association and end up and establish these causal relationships down to the biological phenotype, right? So it would, realistically take us many, many decades, even for type 2 diabetes, to go through these, finding the underlying biological processes that are connected to genetic variation. So how can we, move from this kind of linear, one locus at a time, to a more accelerated type of framework? And I want to just quickly mention two confounders that have been kind of, limiting the community to move from the era of genetic discovery to mechanistic discovery. As we all know, I don't have to tell that this audience, GWAS, is of course coming up with signals that are inherently limited by the haplotype structure of the human genome. The majority of these signals, map to the non-coding part of the genome. We have beautifully seen data from Gosia tackling that. But still, there's a lot of work to do. And the second confounder that we have been particularly interested in, and I love Dave's talk, as I always love your talk, Dave, if you are online, in contrast kind of to principled omics type of transcriptional and epigenomic profiling technologies that get you to transcriptional signatures and epigenomic signatures. When we think about cellular phenotypes in the context of natural genetic variation, we are typically dealing as a community with not only very sparse data types, but also heavily hypothesis-driven assays, right? So we are looking at, for example, here an insulin-stimulated glucose uptake gives you univariate data output. It's very sparse data. For the FTO locus, as an example, we went one by one through every single biological process we could have thought about until we ended up identifying mitochondrial content being under the genetic control of the variation. Similarly so in all of the other single variant to function studies. So currently, identifying cellular biological function is a non-systematic and labor-intensive effort, so there really is a need to develop higher dimension readouts that extract richer representations of cellular phenotypes, but also go beyond only capturing morphology, which is not directly relevant to disease in many cases. So with those two challenges combined, we kind of aim to address them in one of the projects that I wanted to briefly introduce, and with the aim of accelerating variant to function studies. And that is, slightly different from the typical single locus variant to function approach, where here, rather than dissecting each individual genetic risk loci one by one, we are aggregating here genome-wide genetic variation into polygenic risk scores. So we have composite scores at hand, and then we use these scores to select adipose-derived mesenchymal stem cells that are fully genotyped, of course, isolated from donors of a given percentile of the polygenic risk score distribution for a given metabolic trait of interest, to extract from these adipocyte information out of microscopic images, and thereby identify the cellular programs that associate with the polygenic risk score, and start mapping the molecular heterogeneity that we are observing in type 2 diabetes. So very quick introduction of how we have been approaching this. This is work by a fantastic former graduate student in the team, Sophie Strobel, and a former postdoc, Samantha Laber. So and actually a whole village we have multiple team members here who have been pushing this work heavily forward over the last years, and involves an entire village in the team, I guess. So we have developed this metabolic disease-oriented image-based phenotypic profiling tool called Lipocyte Profiler. Lipocyte Profiler allows to extract these rich quantitative high-dimensional representations of morphological and cellular programs that are related to metabolic disease from biological images at the single cell level. We have heard a lot about the modeling aspects of Dave earlier. I'm only going to show you a few slides on applying it to adipocytes. We have applied it in the context of hepatocyte profiling as well, skeletal myocytes, cardiomyocytes, endothelial cells with great outcomes, but I'm not going to go into this today at all. So Lipocyte Profiler very briefly multiplexes 6 dyes in 4 channels, and the dyes that we chose are BODIPI, which stains the intracellular lipid droplets composed of neutral lipids, MitoTracker Red, which we know and have validated to correlate directly with the mitochondrial membrane potential, so it's not only a morphological readout, but actually a direct readout of the activity of the mitochondria. We have Höchst, of course, for the DNA. We have phalloidine for the actin cytoskeleton, wheat germagglutinin for the Golgi and the plasma membrane, and cyto14 that stains the nuclei and the cytoplasmic RNA. Now, this tool, Lipocyte Profiler, quantifies 3,005 non-redundant features across 4 channels, differentiating the organelles, as you can see here in this pie chart on the left side. And you can also see that these features map more or less equally to, the different channels, right? So here, while we had previously, were imaging cells throughout differentiation, for example, and had a single data output of lipid accumulation, now we are looking at 741 non-redundant features that are informative for lipid traits, which in adipocytes can be fusion of lipid droplets to larger lipid droplets, location of the lipid droplet close to the mitochondria, or other phenotypes. So I don't have any time to show you the whole benchmarking of the tool and CRISPR perturbations, etc., that we did to validate it, and want to jump directly into one application where we profile adipose-derived mesenchymal stem cells from subcutaneous and visceral adipose tissue. These are pairs, so we have in total from more than 500 donors these pairs of subcutaneous and visceral primary adipose tissues. And we profile them across the score distribution for type 2 diabetes and obesity-related traits, and then extract these deep cellular phenotypic information out of them, along with, which you will see a little bit of data later on, transcriptional profiles as well, to identify these genetically anchored cellular signatures, right, and start learning about the relationships between the polygenic risk scores and cellular functions that underlie the pathogenesis of metabolic disease. So type 2 diabetes is highly heterogeneous. We, in total, constructed 13, genome-wide polygenic risk scores. I'm only going to mention one of the results of one of the polygenic risk score effects that we looked into, just because it is a nice fit for the pitch we have later, for no other specific reason. We have been working with Jose Flores and Miriam Adler based on their clinically informed partition polygenic risk scores to construct polygenic risk scores that are informative for the polygenic contribution of lipodystrophy, which basically signifies insulin resistance in the context of low BMI and an unfavorable fat distribution. Unfavorable fat distribution meaning a depletion of subcutaneous adipose tissue storage capacity and an excessive amount of visceral adipose tissue. So we have profiled and imaged a set of 71 samples from the across the PRS distribution for lipodystrophy, including both the visceral and the subcutaneous cells, capture differentiation dynamics, 4 stages of differentiation, and it seemed, it seemed promising to us that when we compare the differentiating subcutaneous and visceral cells, we observe highly adipose-depot specific effects. Just in terms of the color coding, very quickly, the red that jumps in your eye is informative for the mitotracker deep red channel. The yellow is the phalloidine for the actin cytoskeleton, and the green is Bodipi for the lipids. So what we are observing are highly depot-specific effects, which you can appreciate, and we also observe that these, cellular programs under the polygenic control of the lipodystrophy cluster seems to follow a certain differentiation dynamic with the highest effect in the fully differentiated state, right? Where the adipocyte is metabolically highly active. So for subcutaneous differentiated adipocytes, we observe a cellular signature that is characterized by features informative for mitochondrial impairment and actin remodeling processes, and decreased lipid accumulation. And for visceral adipocytes, we actually observe an increased lipid accumulation and a heterogeneity of the lipid droplets in the cell. I'm also only going to focus now on the subcutaneous differentiated adipocyte effects, where you can see here on the bottom of the slide, when we look at transcriptional signatures, basically assayed at the exact same stages of differentiation from the same individual, we see the same type of differentiation dynamic that the transcriptional signatures follow as the image-based profiles follow. And now genes that are correlating with the polygenic risk scores, they appear to be enriched in processes involving fatty acid catabolic processes and respiratory electron transport, pointing again to the mitochondria with complementary evidence, right? Now, what seems striking to us is when we look at the correlation signatures between these monogenic markers of partial lipodystrophy. There are multiple monogenic markers, PPA gamma, for example, ACT2, LIP, paralipin, SIDEC, or laminin A. And the image-based features, we find that these monogenic genes, they seem to either cluster by mitochondrial features, and I can tell you the features are matching the features that we are observing for the polygenic contribution to lipodystrophy, or they are matching, again, the actin remodeling feature or signature. So Felipe, a wonderful postdoc in the team who's here today, somewhere, looked at the individual features, and I just want to highlight this very quickly in terms of what is driving this process. The features informative for the polygenic risk score effect were primarily intensity features of the mitochondria. and a few granularity features. And the mitochondrial intensity features, they were actually all matching to the feature class of the edge. So there seems to be a hyperactivity of the mitochondria at the edge of the cell, which seem to be really interesting to us, as well as a perturbation of the actin cytoskeleton and a decreased lipid accumulation that I mentioned before. Now, when we look at individual, a little bit deeper, we can actually map, this is just an example where we are looking at the transcriptional effects of one of the top scoring genes, a gene that is involved in the mitochondrial fragmentation process, which maps the image-based feature mitochondrial granularity that is informative for mitochondrial fragmentation. And then you can see these top scoring features of the mitochondrial intensity at the edge of the cell. Now, what then Phil, a fantastic grad student in the team who's also here today, what he did in a really heroic effort, I would say, is systematically model the insulin responsiveness in the cell by a glucose titration screen, basically an entire different screen from the lipodystrophy polygenic risk score effect. And the top scoring feature that he models, the relationship with insulin resistance, seems to be the mitochondrial edge intensity feature. So we are super excited about this and starting to, find, validate this kind of cellular phenotype of highly hyperactive mitochondria at the edge of the cell that are leading to insulin resistance as a phenotype that is, of course, relevant for type 2 diabetes, right? Part of the validation, I would just want to include one slide here, is from our single locus variant to function work at the locus on chromosome 2 that we connected to its effector gene COPLA1. When you look at the haplotype effect, that's the lower left bar plots, you see this kind of nice dosage effect across the differentiation trajectory for the risk haplotype, increasing the mitochondrial edge intensity relative to the non-risk haplotype. When we now perturb the COPLA1 effector gene itself, we observe the same feature. I'm not showing this here, but we also really can show that the subcutaneous adipocyte is resistant to insulin in an insulin-stimulated glucose uptake assay. We also work with Marcelo Nobrega. Marcelo, or Deborah and his team, created the COPLA1 knockout mouse. And, very quickly, overall notion here is that the knockout mouse appears to recapitulate the phenotype that we are observing at the organismal level. Now, I want to close this with images that are computationally averaged across thousands of single cells from individuals that sit at the top and the bottom of the polygenic risk score distribution for lipodystrophy. And what you can appreciate is a very, very stark difference, right? You see this hyperactive or this very, very strong mitochondrial signal in subcutaneous adipocytes from high polygenic risk individuals relative to low polygenic risk individuals. So there's a lot of biological information to extract. What we now have to do is to, add in many, many more markers, many, many more information features to really close this gap between genetic variation, polygenic risk scores, and their underlying biological function. So I want to, shout out to the lab. Hesam Dashti has contributed tremendously to this project, as well as has Sakit and Yuang, among many others. I want to have a huge shout out to Cecilia, who has been a wingmate for this project over the last years, and I love working with you, as you know. And Carpenter, dear colleague of ours, who has been working through this project. And I mentioned Miriam and Jose and Shema, of course. And really want to thank everyone for your attention, and happy to take questions.
One in the room over there.
Time for a question there.
Jordi Merino, University of Copenhagen. Thank you for a very, very nice presentation. I really love it. So one fundamental question that I have is about the use of polygenic scores for inferring causal mechanisms, identifying cellular processes, and all this amazing stuff that you show. The story makes a lot of sense, and it's beautiful. But my question is, or my observation is that the way that people can be in the high or low distribution of the polygenic scores may reflect also an heterogeneity or combination of mutation, signal defects. So to me, it's less clear that these groups of individuals at the higher, lower distribution of the polygenic score share exactly the same similar mechanism leading to increased diabetes risk. So would you like to comment on that?
No, that's a great question, though. I just want to clarify. The only slide that I showed that was selected from the tail ends of the score distribution are the images, the ground truth images. Everything else was regression, including across the score distribution, right? So it's not a comparison of the tail ends of the score distribution.
Yeah, but these distributions reflect different mechanisms, right?
Yeah.
Right. So how you go from this heterogeneous?
Yeah.
Sorry if I'm allowed to follow up. So these tails reflect different genetic variants, and the way that people can be scored as a very high or very low might be different, might reflect different underlying processes. So my observation is that maybe your results would be even strong if you were able to better quantify or better categorize these subtypes of individuals at higher, lower genetic risk.
Yeah, no, that makes sense. I mean, these profiling data are from 26 donors in total with the pairs of subcutaneous and visceral isolated cells. What you're referring to is absolutely, I agree entirely, though needs, lots more sample size to get to that. Lots higher sample size, not more.
Carl Anderson, welcome, Sanger Institute. Two fantastic talks. My question is actually for Gosia. And Gosia, I can't help but feel I've probably asked you this back at Sanger before.
Oh, no. Are we going into this again?
It concerns the dynamic EQTLs and which piece of the EQTL you think is dynamic. So obviously you have expression changing over time, and when things are more highly expressed, you have more power to detect EQTLs. Do you think that these dynamic EQTLs are really gene expression changes exposing EQTLs where the beta is essentially fixed over time? Or do you feel that the beta is changing over time, or is it a mix of both? And if so, do you have a sense of which is playing a bigger role?
Yeah, I think it could be a mix of both. I mean, we detect very different effects, these dynamic effects. We detect effects where you can see stable effects across multiple time points of activation. We've got these clear peaks. We did have this conversation, Carl.
We're a little bit really behind schedule, so I will ask you to have this conversation over a glass of wine.
I just wanted to make one point. I think, we tend to think about cell function based on high expression of certain genes. So knowing at which point you have this high beta is really important for biology. And we can get into details of a drink later on.
Okay, let's move on with the pitches because we're really a little bit behind schedule. If that's okay, thank you. I know there was a question.
Can I still ask my question?
Yeah.
Okay, good. So a question for Melina. So thank you very much. I really enjoyed your talk. So I was wondering if you can comment on this idea of the partitioning of the genetic architecture into different processes and our ability to do it across all diseases.
I think we have the ideal candidate to do that in the audience, even if you want to take that. So Miriam in José's lab in her time has, this is not our algorithm, our technology, but similarly so as Mark has done with Anuba Maharan, asked the question of subclustering of type 2 diabetes. And for Miriam, she used clinical traits like lipids, triglycerides, BMI, waist-to-hip ratio, etc., in the context of the type 2 diabetes signal and clustered them through a Bayesian framework in the relative, in 5 clusters initially, and she's currently at 13 clusters as far as I know, with increasing sample size on the GWAS, as well as increasing clinical traits that she's folding in. Do you want to expand? Great. Yeah. Or Mark maybe. Mark can also expand.
Just to say there will be more opportunities to ask questions to Gosia and Melina in the discussion panel at the end of their final talk. But in the interest of time, I think we'll move on to letting them present their final pieces for us today.
All right. So it's a great pleasure to be here, and we hope we will get you as enthusiastic about this proposal as we are. So the past 2 decades have been transformative for human genetics. Mapping variants associated with diseases and quantitative phenotypes has become a routine. At the same time, large-scale coordinated efforts generating resources looking into gene expression regulation across tissues and cell types have been providing links between disease associations and effector cell types and tissues. While these efforts have been valuable and have provided important insights into disease biology, they have been hugely limiting in delivering insights into the mechanisms through which disease variants and genes regulate cell function, the actionable biological processes that offer opportunities for therapeutic interventions. So, on one hand, thousands of disease-associated variants, and on the other hand, resources enabling us to make partial links to disease-causal genes and cell types. We know there are different disease underpinnings, but we struggle to conclude which biological processes they regulate. It's a bit like we're staring at cars of different make. We can tell that the cars are different in size, color, type of tires, or the size of the engines. But the issue is that we're trying to infer the impact these car parts have on the performance of the cars by looking at them statically. And until we actually put the cars through the road test, we can't make causal links. Does a type of a tire, the size of an engine, or a color of a car impact the car's performance? How much do these elements impact acceleration or fuel economy? Is the car's color correlated with the size of the engine? Until we start looking at the mechanics of cellular processes dynamically, dissecting them into individual cellular components, the parts of different engines that make up for differences in cell types and their effector functions, we will not be able to conclude the role genes play in regulating cell biology and their impact on disease. So to bring it into a biological context, this is where we are now. We are making inferences about molecular underpinnings of biological processes by looking at a snapshot of a process. Like looking at a T cell interacting with a cancer cell, we profile the cell at one time point, typically in a resting state, and measure the endpoint of the process, successful or unsuccessful cell killing. Such snapshot approach to biology misses out a whole complexity of changes in molecular and morphological features that take place in the cell for it to execute this process. And we don't know which of these are important in regulating this process. What we should be doing instead is looking at the dynamics of cellular processes, the mechanics of T cell migration to the target cell, how strongly it attaches to it, what is the impact of actin remodeling in this process, the metabolic activity of a cell and how it impacts on the secretion of toxins that eventually kill the cancer cell. And which of these processes are important? How are they genetically regulated? And what are the links to the diseases? This is our current state of looking at cellular processes in a very fragmented way. We may use transcriptomics, proteomics, or cell imaging to correlate molecular layers with a cellular process. In practice, RNA-seq is the current standard in the community, but the other modalities are not. Rarely do our approaches multiplex the assays and do it at scale with deep dynamic sampling and with single-cell resolution. Well, in fact, there is no cellular phenotype that is mapped at these depths, so we really have no clue what are we dealing with. So imagine a resource that would comprehensively and systematically multiplex these different cell readouts across different cell types in different conditions, studying various cellular processes across thousands of individuals from diverse ancestral groups. A resource that would be powered to map the genetic regulation of cell morphological changes across different cell processes and map the effects of transcriptome and proteome to infer causal relationships between these modalities and cellular processes. A resource that would ultimately deliver the links between disease genetics and disease biology for actionable therapeutics. So the result of our working group is a project proposing a community-driven effort to map genetic variation underpinning cell functions.
Yeah, thank you so much, Gosia. So center of the pitch really is biological cellular processes, as Gosia has highlighted. And I guess, you know, the notion we are discussing here in the ICDA for a few years now really is that our community has incredibly well figured out to link genetic variation to diseases and traits, right? And I guess the success of the, you know, genetics revolution was driven in large pieces by introducing transformative technologies to the community that then were taken up by the community and really harmonized, implemented, and democratized across the entire community, and kind of enabled the era of, you know, genetic discovery. So, you know, similarly so, the era of mechanistic discovery might require, or will require, as we think, a similar transformative technology informing cellular processes, a tool that basically can be as standardized, democratized, implemented in our community, and really, once the community stands behind it, change our foundational understanding of the biology of disease, right? So with the different talks we had today, with the V2F work that has been beautifully going on in the community, we have this playbook or a blueprint for how to go and you know, trace genetic variation effects that cascade kind of to, you know, all levels of cellular biology and propagate across these different layers in the network, right? So when we look at the cell seems very, very complex, as biology is, and it, you know, is this kind of complexity, or the combinatorial challenge is captured in this kind of layered framework that you're seeing here. But there's logic to it. And we can ask, what are the principles and the rules of such cellular programs? And I guess, you know, a generic model, of course, that we can start with is knowing the hierarchical relationships between molecular layers from the dogma of biology, where we have really an intuition that information flows across the molecular and cellular networks in the cell, from DNA to RNA and protein, and then, you know, impacts morphological and cellular phenotypes as kind of our natural, you know, kind of integration points of the effect of many alleles and many genes that are impacted by genetic variation associated to disease. We also know that cellular networks vary across cell types and states. They are highly dependent on stimulation of cells with you know, physiological stimuli or drugs, and operate in a very dynamic manner, as Gosia has beautifully shown. I've provided a few examples as well. So we envision to study cellular programs with resolution for single cells to capture the stochastic process, and use complementary high-dimensional readouts that are really poised to capture these, you know, gene regulatory circuits, the protein circuits, and the cellular networks across these cell state transitions. And once, you know, they can be integrated, really border the different molecular and cellular layers of information in the cell. So our pitch is really to envision the next phase of human genetics, where the community comes together to develop, benchmark, and scale a foundational tool coined CellMapper to deeply and, you know, reproducibly profile cellular processes by capturing complementary phenotypic information across many cell types at population scale, to power the discoveries of cellular processes that underlie these disease association, and really, you know, introduce a new variant to function paradigm that we think could capture the causal relationships between genotypes and phenotypes through mechanistic models, really, of cellular programs. So what does such a tool have to have as characteristics? Such a tool, if we want it to be transformative, has to be, you know, it has to be very robust. It needs to be compatible across cell types, such that Carl, you were there sitting earlier on, can come with an IBD interested epithelial sample and can be pulled through the tool. I see Tune beside Carl, interested in neurons. We see this a community effort with the, you know, leaders in the field, as all of you are, coming with a cellular model that you have your expertise in, and help, you know, optimizing such a tool. We want to be, you know, the cell types that we are envisioning currently to be plugged in are immune type of cell types, iPSC, as well as primary MSC-derived model systems. Such a tool also needs to be scalable, of course, to allow for mapping cellular progresses across thousands, actually tens of thousands of individuals, as Gosia will tell you later what our plans are. It needs to be highly reproducible, needs to allow for harmonized matched data creation, implementing these complementary readouts that are, you know, captured or poised to capture the dynamic cellular processes. And kind of the best analogy that we could think about is a dispersive prism, right? Where in this case you see this triangular from connecting genetic variation to deep cellular profiles and to diseases, and similar to such a dispersive prism that breaks up the light kind of into its constituent colors. Here we are breaking up the cell into its highly granular biological processes and inform how genetic variation can affect disease. Now, how do we do this? We have been discussing this quite a bit. Here's a high-level overview of 3 different phases that we are envisioning for developing the CellMapper paradigm. We have a benchmarking phase that runs over 3 years, proposed 3 years, a scaling phase and a mapping phase, scaling for 2 years and then mapping for 3 years. You see the number of individuals for screening purposes increases drastically over these 3 phases and ends up in 10,000 individuals at the last, at the last stage. Of course, simultaneously, so we have to find a principled approach to kind of subset stimulatory condition, differentiation time points, and cell types that can be pulled into the characterization of 10,000 individuals. And Gosia will come to this later. For now, I'm going to focus on the benchmarking phase, where we are really, ideally, we would do that in any kind of cell type we can think about, which is not practical, of course. So we would prioritize, including the community, 8 to 10 cell types for the initial benchmarking phase, and would systematically probe the cellular models in response to a broad set of disease-relevant stimulatory conditions, diverse time scales to test the response effect of stimulation in the context of time points and differentiation with the single technology, with this tool that is calibrated well enough to really capture cellular processes across different cell types. Why do we think, as an example, just provide an example here, why do we think that a single tool can inform different cell type cellular programs? Here's an example for high content imaging based mitochondrial features, as you have seen, for example, in our work on the lipodystrophy polygenic risk score, pointing to mitochondrial hyperactivity and fragmentation as related to insulin resistance, as well as lipid metabolism and beta oxidation. The mitochondrial intensity for T cells is informative for T cell activation. For neurons, it's actually informative for the differentiation process, as well as axon branch maturation, as an example. Another example is the mitochondrial count, which is the biogenesis of the mitochondria, that is informative for browning, thermogenesis, and adipocytes, while it is, in T cells, informative for memory, T cell formation, and in neurons, synaptic activity and synaptic plasticity. Coming back to this example that I've showed you before, where we are looking at a genetic effect, in this case a score, we can place certain genes into a pathway informed by the high content imaging, extract these cellular programs that are under the polygenic control, which relate to insulin resistance. Now, how do we actually measure a multitude of markers at the single cell level comprehensively to map these cellular processes? There are 2 technologies in discussion as part of the CellMap team that we think are in the sweet spot in terms of scalability, feasibility, and capturing complementary phenotypic information. And when I say feasibility, then we have excluded certain tools because they might not be amenable to certain cell systems like immune cells or iPSC-derived models. So the 2 systems that we think make sense is CytSeq, that measures single cell gene expression profiles, as well as single cell level surface protein abundance, really for functional markers at the scale of hundreds of multiplexed markers that tell us about cell signaling really and cell identity. Such as increased expression levels of the CD69 markers and early activation markers of T cells, as Gosia has beautifully shown, or in adipocytes or skeletal myocytes. It would be, for example, the glucose transporter 4. Once it's at the plasma membrane, it's a direct proxy of the actual insulin responsiveness of the cell. So when we look at the dimensionality of, including all of these features, the feature space is really large. We are covering, quite complementary information of tens of thousands of features going to the gene expression level, hundreds of features mapping to the surface protein, cell signaling information, and then thousands of features that we can get from the image-based profiling, where here we are thinking about combining morphological markers with markers informative for cellular processes. So, this way we can start mapping how cells behave in the context of cell activation and differentiation time course. And I've, as I've shown you today with the lipodystrophy polygenic risk score effect, when you remember this kind of differentiation trajectory that we found both for the image-based profiling as well as the transcriptional profiling, these type of data sets in the benchmarking phase can help us to subset down differentiation time points. For example, we don't need to interrogate every single differentiation time point in the adipocyte differentiation based on, thorough benchmarking experiments of stimulatory conditions. Now, I think I'm running slightly out of time. We really, with this tool, we aim to generate a deep reference map characterizing the responses to various stimuli, understand how cells respond dynamically, right, and really provide concrete cellular biological phenotypes that we can connect to genetic variation. Just closing here, we come back to this PRISM analogy. We really have, this genetic variation. Ben pointed this out beautifully this morning as a shared coordinate space, which means that the genetic regulators here of the, cellular traits really can be linked to human disease through the identification of these, shared genetics. And, from the gene dosage response curves, we know that when multiple distinct variants are mapped to cellular phenotype, it really enables the direct evaluation of whether this phenotype is on the causal path. So the benchmarking phase will give us a set of stimuli, stimulation time points, differentiation protocols and cell types, an established cell mapper workflow, and a standardized pipeline that is reproducible across labs, and a deep reference map. And it really is aimed to catalyze the community and exchange knowledge and develop the CellMapper tool for population scale mapping of cellular functions.
So the benchmarking phase will deliver a first-of-its-kind map deeply and dynamically characterizing molecular and cellular traits in the context of disease-relevant cell types and cell phenotypes. It will also inform the next phases of the project towards delivering the cell map. Following benchmarking, the scale-up phase will take a funnel-down number of cell types, stimuli, and time points, and apply the cell mapper to profile selected cells across hundreds to 1,000 individuals. The goal of the scale-up phase is to assess the feasibility and variability at a sufficient scale to informal initial morphological and cellular QTL mapping. And the outcome of the scale-up phase will be the final selection of assay and cell types, a combination that will be ambitious but practical for the mapping phase of the project. The aim of this last mapping phase of the project is to apply the cell mapper across thousands of individuals to deliver QTL maps of cellular morphological, transcriptional, and proteomic readouts. We propose 10,000 individuals from diverse ethnicities to ensure statistical power to map the impact of genetic variation on regulation of cell traits. Project of this scale will generate a wealth of data and will require data coordination and infrastructure, for example, adapting a model similar to the Human Cell Atlas. We also envision a community-driven effort to support the analysis, develop data standardization practices, frameworks for QTL mapping, and integration of data across modalities and with disease genetics, as well as data visualization. Where suitable, these efforts should leverage existing databases, as well as drive the development of new resources. So finally, we will capitalize on sample recruitment from biobanks with deeply phenotyped populations. We have had preliminary discussions with UK Biobank and have the enthusiastic support to tap into a prospective recruitment, which would specifically upsample individuals from across genetic ancestries to increase the diversity of the cohort. The sampling would include a pinch needle skin biopsy and a large blood draw. The skin biopsy provides a source of fibroblasts, which can be reprogrammed to iPS cells, while adipocyte tissue can be used to isolate mesenchymal stem cells, giving us the benefit of differentiating primary stem cells into different cell types, as Melina was beautifully showing us earlier in her talk. And then blood would be the source of primary immune cells. And we have also had initial discussions with the New York Stem Cell Foundation, which offered to support automated iPSC derivation that their facilities are very well set up for. So ultimately, CellMap CellMap, through its different phases, will have a far-reaching impact through a community-driven effort working with domain experts. We will deliver a benchmark and standardization of cell differentiation and stimulation protocols. We will establish the CellMapper tool and apply it at scale to deconvolute the mechanistics of disease-relevant cellular processes to individual molecular modalities and identify causal relationships between them. This deep reference map characterizing the genetics of cell function will provide a roadmap for moving from disease associations to disease biology. And I'd like to end by acknowledging all of the CellMap working group team members who have contributed towards developing this proposal. We are very enthusiastic about this proposal. We think it has a transformative impact, and we hope that the community will see its impact as well and will feel supportive as well. Thank you very much.